Hello guys, so today we're looking at our cell division and cell cycle. So today is part 1. So our objectives today are look at the cell theory, prokaryotic cell division, and eukaryotic cell division, but we'll get to mitosis only today. So the cell theory. The cell theory is one of the fundamental principles of modern biology. The cell theory has 4 points. So 1, all organisms are composed of cells or cell products. 2, all cells in an organism have the same DNA and the number of chromosomes unless they are sex cells. 3, the chemical reaction characteristics of living systems occur within cells. And 4, all cells arise from pre-existing cells. So the genetics are passed down. So here's a broad overview of cell division. So cell division essentially is how we reproduce. So cells reproduce via cell division. So the key roles of cell division are reproduction in unicellular organisms, growth and development in multicellular organisms, and cell and repair in multicellular organisms. So we can see different examples here. So cell division is the process by which a single cell becomes 2 daughter cells. So essentially it's just this cell becomes 2 more cells. And this can repeat again and again in certain divisions. Cell division involves the division of the nucleus and the cytoplasm. So we have karyotype, which is division of the nucleus, and cytokinesis, which is division of cytoplasm via the formation of a cleavage furrow, which we'll discuss more later. The requirements for successful division are 1, full complement of DNA must be distributed to the 2 daughter cells from the single parent, so nothing must be left out. The parent cell must be large enough to divide into 2 and still contribute cytoplasmic components to each of the daughter cells. So methods of cell division. So we have binary fission. So in binary fission, this is asexual reproduction, where a new copy may be identical or have mutations. This occurs in prokaryotes like bacteria and other single-celled organisms like amoebae and paramecia. We have mitosis, which occurs in somatic cells and produces exact copies of parental chromosomes, so they're identical. Well, meiosis, which makes sexual reproduction possible. So meiosis is involved in sexual reproduction. Meiosis produces daughter cells, which are called gametes, with half the number of chromosomes compared to the parent cell. They're not exact copies. So first we'll discuss binary fission, which occurs in prokaryotic cells. So binary fission, so most bacteria, which are have one circular chromosome. We can see here that circular chromosome. And it can be highly folded and coiled, so, like these examples. So there's a replication of DNA, which the cell increases in size. There are 2 important regions on the DNA, the origin of replication and the terminus of replication. So essentially that's the start and the end. We have DNA segregation, which we can kind of see here, where 2 origins are attached to the plasma membrane, move apart. Then we have cytokinesis, which is the pinching of the plasma membrane and the synthesis of 2 cell walls. So we'll go through this figure here in a little more detail, similar to what I just said, but slightly more detail. So first, we have the circular bacterial DNA molecule. It's attached to the biproteins in the inner membrane. So we can see here it's attached. DNA replication begins at a specific location and proceeds bidirectionally around the circle. So here's a circle, it's going bidirectionally. The newly synthesized DNA molecule also attached to the inner membrane near the attachment site of the initial molecule. So we see they're still attached, but we have newly synthesized DNA. As the replication proceeds, the cell elongates symmetrically around the midpoint, DNA separating, and then there's separating of the DNA attachment sites, where we have DNA segregation. Then we have cell division begins with the synthesis of a new membrane cell wall. So we see the formation of the wall occurring, and cell material at the midpoint. Then there's the continued synthesis and completes the construction of the cell wall, and then we have cytokinesis, which separates the daughter cells. So again, good thing just to keep looking at the figures is they provide a good visual learning tools. So in eukaryotic cells, we have mitosis, but also meiosis for sexual reproduction. but we'll focus on mitosis for now. So the result of mitosis is that we have two identical daughter cells with the same number of chromosomes as the parent. So usually we have 46, and then these daughter cells will also have 46. As compared to meiosis, where we have 46, and the daughter cells will have, but we'll get to that next time. So mitosis still involves the general steps seen in binary fission, but more complex than prokaryotics. So something key in all cell division is the cell cycle, which is something I definitely recommend looking into. I won't go into too much detail, but it's something that's quite important. So here we can see there's several phases. So cell division proceeds through a number of steps that make up the cell cycle. There are two distinct stages, the M phase, interphase. So the M phase is the time during which the parent cell divides into two daughter cells. Then we have the interphase, which is the time between the two successive M phases, which also includes the G1, S, and G2 phase. So here we see our M phase where you have mitosis or cytokinesis. We also have something called the G0 phase where cells that are not actively dividing, for example, a lot of our neurons don't divide later in life. So these don't go through cell division. Then we have our G1 gap 1, our S phase where we have DNA synthesis, and our G2, which is gap 2, and we also have this interphase, which again is the time between successive M phases. Again, these are the distinct stages, not all the stages. So what about the G0 phase? Like we said, not all body cells actively participate in the cell cycle. Many cells pause between the M and S phase. So G0 cells remain active in other ways. So example would be liver cells that metabolize and detoxify. They're working, but they're not being divided. And again, nerve cells enter the G0 phase permanently. It cannot be divided. So this is when you hear people say, don't hurt your brain cells. If you lose your brain cells, they never come back. That's essentially due to this permanency in the G0 phase. So mitotic cell division. So just to overview, we have our chromosome structure. So in eukaryotic cells, DNA is organized with histones and other proteins into chromatin. So chromatin can be looped and packed to form chromosomes. So you've probably seen these chromosome things before. So here we can see double helix DNA. We have our histones, different kinds of histones. These are formed into the chromatin, and then this chromatin is packed. You can see here you get the sizes. They're packed into this this new chromosome. So here we see a metaphase chromosome. So we also have something called a karyotype. So here, this is kind of a display of all the chromosomes in the person. So as we said, each chromosome has 23 times 2, essentially. So here we have a karyotype depicting the person's chromosome. So here we see the X, which is a sex chromosome. So here's a portrait formed by the number and shapes of chromosomes. So that's karyotype. Is representative of as a species is called as karyotype. So in humans, we said we had somatic cells where we have 46 chromosomes, and gametes have 23 chromosomes. So those are the daughter cells. So the human karyotype, there are 22 pairs of homologous, so again, pairs. So that's how we get to 46, 23 times 2. We have the homologous chromosomes, which is the autosomes. So all non-sex chromosomes are considered autosomes. Then we have one pair of sex chromosomes, X and Y. So again, 22 times 2, we have our 44 autosomes, and then plus 2 sex chromosomes, we get our 46. And again, 46 is in our somatic cell, where 23 is in our gamete. So homologous chromosomes. So what this means is that two of the same type, if we have two of the same type of chromosome, this is a homologous chromosome. So each homologous chromosome carries the same set of genes, and one is homolog is parental and the chromosome, and the other homolog is maternal. So again, here's our chromosome. One is maternal, one is paternal. But they have identical functions. They're the same type. So the number of complete chromosome sets is called ploidy. We can have haploid versus diploid. So homologous chromosomes and sister chromatids. So for cell division to proceed normally, every chromosome in the parent cell must be duplicated. So each daughter cell gets the full set of chromosomes from the parents. After duplication of DNA, each chromosome is made up of 2 sister chromatids. So that's important. So we'll go through this more. So sister chromatids are held together at the centromere. So essentially, the centromere is just a little connector. So here, note to count the number of chromosomes, count the centromeres. So again, we have our 2 separate centromeres, homologous chromosomes, so one would be maternal, paternal. They enter the S phase. Then we can see here, they now become, we have 2 sister chromatids. And again, we have the centromeres. So mitosis. So this is, again, we talked about our cell division. So one way to remember this right off the bat is called pomadi. So again, we'll go here, but we sometimes add the one where it's interphase. But essentially interphase is kind of happening the whole time, but that's a good way to remember it. So we have our prophase, a prometaphase, our metaphase, anaphase, and telophase, and then interphase. So remember, in interphase, you cannot distinguish specific chromosomes because they're all long and thin. So step 1 is prophase. So just keep this in mind. So sister chromatids are joined together by cohesions. So step 1, prophase. The chromosomes condense and the centrosomes radiate microtubules and migrate to opposite poles. So here we can see here, moving to opposite poles. That's our centrosome. And we have our mitotic spindles. Then we can see in here, we have our chromatin fibers and a nuclear epitope. So as we see this is happening, we see our chromatin is condensed into those chromosomes. Then step 2 was our prometaphase, where we have the nuclear membrane breaking down. So we can see here, we have sister chromatids and a nuclear membrane starts to break down in our chromosomes. So these centrosomes and their microspindles are kind of pulling these apart in an attempt to form 2 cells. So these spindles attach to chromosomes at the kinetochores. So the kinetochores are where the chromosomes, the spindles attach. These microtubules polymerize and depolymerize. So what do kinetochores look like? So again, just to orient us, we've got a centromere, kinetochore, microtubule. So here's our microtubule. We can see this kinetochore protein. So these kinetochores are protein complexes. Each kinetochore is associated with 1 sister chromatid, so that's why we got 2. And there's attachment site for a single microtubule. So see here, microtubule and the chromatids. So now on to step 3, metaphase. So in metaphase, there's the alignment of the chromosomes in the metaphase plate. So metaphase plate is kind of essentially this area where they're going to line up. So we saw originally that over here, they're all oriented in different sides, different angles, etc. Now, through metaphase, they've become oriented and they're going to be essentially lined up single file in that line where they can be split apart. So again, metaphase, chromosomes aligned in the center of the cell. We can see here spindle pores, which are releasing those tubules to attaching to the kinetochores like we talked about. So next we have the anaphase, where the sister chromatids separate and cohesion proteins are cleaved. These non-kinetochore microtubules, so these are non-kinetochore microtubules, and these chromatids become chromosomes. So again, there's equal segregation of the chromosomes, so it's not unevenly distributed. So we can see here it's even. So in anaphase, the sister chromatids, which become individual chromosomes when the centromeres split, separate and travel to opposite sides or opposite poles. So next we have telophase. So in telophase, the nuclear envelope reforms and the chromosomes decondense. So now we start to see the division starting to take place. So those chromosomes packed into those things go back to their chromatin form and see how they become all spiraled out again into the nucleus. So next, in the humans, we have cytokinesis, which essentially forms this contractile ring around the two cells and essentially cuts it. Here which is formed, so we have actin in a contractile ring causing this cleavage furrow, and it splits the cells. While in plants, we have cytokinesis, where we can see here that we have phagomoplast and vesicles and cell plate, and this cuts the cells. So again, the result is that two diploid cells that are genetically identical to the parent cell are formed. So again, mitosis, identical to the parent cell. The reason this is emphasized so much is the comparison when we get to meiosis. So again, that's our very brief overview of our prophase, prometaphase, and our metaphase, anaphase, and telophase. So again, here's our recap, our 5 different stages. And then again, next we'll get into meiosis, which is involved in daughter cell formation. But again, I hope you enjoyed this brief, very light discussion of this, because this could also go much deeper in all the different proteins and all that stuff like that. But yeah, this is, again, a basics needed for cell division, which comes up quite a bit, especially in different diseases. And we'll see this in our pathology lectures as well. Anyways, take care. See you next time.